In Chapter 1, we were talking about single gene diseases, and we looked at how those diseases are inherited in individual families by analyzing pedigrees. Now in Chapter 2, we're going to continue to talk about single gene diseases, but in Chapter 2, we will be talking about how those diseases are inherited in large populations, that is, population genetics. On your exam, there are certain calculations that you will be asked to perform to predict, for example, the fraction of a population that are carriers for a genetic disease, or the fraction of a population that will be affected by a genetic disease. And in this chapter, we'll learn how to do those kinds of calculations. In this chapter, they will introduce for you two terms that we will use throughout this chapter and actually throughout the whole section on medical genetics. The two terms are genotype frequency and allele frequency. Let's look at genotype frequency first. Genotype frequency refers to the proportion of each genotype in a population. As an example, suppose we were talking about sickle cell disease in a population. In that population, there would be people who are homozygous normal. There would be people who are heterozygous or carriers for sickle cell disease. And there would be people who are homozygous mutant or affected by sickle cell disease. Those represent three possible genotypes in the population. Genotype frequency would mean, in that population, what fraction or what percentage of the people in that population are homozygous normal? What fraction or what percentage of them are heterozygous carriers? What fraction or what percentage are homozygous mutant? So typically there are three genotype frequencies, three genotypes and three genotype frequencies in a population. Now what about allele frequency? The proportion of chromosomes that contain a specific allele in a population. Suppose, again, we were talking about sickle cell disease in a population. We would be talking about the beta globin gene. And in the population, there would be two different alleles for beta globin. There would be normal alleles for beta globin and mutant or sickle alleles for beta globin. Allele frequency would mean, of all the beta globin genes in that population, what fraction or what percentage of those genes are normal, and what fraction or what percentage of those genes are mutant. So typically there are two allele frequencies, two different alleles and two different allele frequencies for most genes. Now in order to illustrate that terminology, let's look at a restriction fragment length polymorphism, or RFLP. A restriction fragment length polymorphism, or RFLP, is a kind of gene that we sometimes call a DNA marker gene. That is, this is a gene that doesn't necessarily have any function at all, is not necessarily related to any disease at all, but it is simply a gene, a piece of DNA that we can identify in the lab by a technique called southern blotting. And in the population for this gene, there are two different alleles, two different variations of the DNA base sequence, such that that DNA in those two different alleles is cut differently by a restriction enzyme, such as EcoR1, for example. And so, for example, in the population, we might have allele 1 of the RFLP gene is cut in such a way that we get the upper band on a southern blot. On southern blots, you should remember that larger pieces of DNA appear near the top and smaller pieces of DNA appear near the bottom of the southern blot. So allele 1 might give the upper band on a southern blot. Allele 2 will have a different base sequence, will be cut differently by EcoR1, and will give a bottom band, a lower band on a southern blot. So two different alleles for this RFLP gene in the population. Now, because we have pairs of chromosomes, each person has 2 copies of each gene, and so in the population, there will be people who have 2 copies of allele number 1. They are homozygous 1-1. We can determine who they are because they will only have 1 upper band on the southern blot. And you might notice that this band is drawn thickly. It is thick, drawn thicker, because there are 2 copies of that band in these people. In the population, there will also be people who have 1 copy of each allele. They are heterozygous 1-2. They will show 2 bands on their southern blot, and these bands are drawn thinner because there is simply less DNA in each one of those bands. And in the population, there also will be people who are homozygous 2-2. We can determine who they are because they will have only the lower band on the southern blot. So the point is, we can use southern blotting to identify the genotype for this gene for any person in our population. In the example in your book, the investigators looked at 100 different people, and they have identified, well, there are 3 possible genotypes in the population: homozygous 1-1, heterozygous 1-2, homozygous 2-2. There are 100 people in this population, and count means how many people did they identify as having each of the possible genotypes when they ran the southern blots. By southern blotting, they found 49 of those 100 people were homozygous 1-1, 42 of the people were heterozygous, and 9 of them were homozygous 2-2. So count means the raw number of people with each genotype. Genotype frequency means what fraction or what percentage of the population is found in each of those 3 possible genotypes. So the genotype frequency for the homozygous 1-1 people would be 49 divided by a total population of 100. The genotype frequency for the homozygous 1-1 people would be 0.49 or 49%. 49% of the population is homozygous 1-1. And then by similar calculations, 0.42 or 42% of the population have been identified as heterozygous. 0.09 or 9% of the population is identified as homozygous 2-2. So these 3 numbers are the genotype frequencies for the 3 possible genotypes. Now that we know the genotype frequencies, can we determine the allele frequencies? Yes, we can. This table shows a calculation for allele frequencies. The first 2 columns in this table are the same as we just saw in the previous table. That is, in the left-hand column, we list the 3 possible genotypes. The second column shows the number of people we found with each of those 3 genotypes. So there were a total of 100 people. Now, since we have pairs of chromosomes and each person has 2 copies of almost all the genes, if our total population is 100 people, in those 100 people, there will be 200 total copies of this RFLP gene in question. And allele frequency will mean, of those 200 copies of the RFLP gene, what fraction of those RFLP genes are represented by allele 1, and what fraction of them are represented by allele 2. The calculation for the allele frequency of allele 1 is shown in red in this slide, and we determine this by simply counting up the number of copies of allele 1 in this population and then dividing by the total number of genes 200. Since there were 49 people who were homozygous 1-1, each of those 49 people has 2 copies of allele number 1. So there are 2 times 49, or 98 copies of allele number 1 in those people. Each of the 42 heterozygous people has 1 copy of allele number 1. So there are 42 additional copies of allele 1 in the heterozygous people. The homozygous 2-2 people don't have any copies of allele number 1. So to determine the allele frequency for allele number 1, we multiply 2 times 49, add 42, divide everything by 200. Our allele frequency for allele number 1 is 0.7. That means 70% of those RFLP genes in this population are represented by allele number 1. And then we can do a similar calculation for allele number 2, shown in yellow. Each of the 42 heterozygous people has 1 copy of allele number 2. That would be 42 copies. Each of the 9 homozygous 2-2 people has 2 copies of allele number 2. It would be 2 times 9, or 18 copies. We grind through all the math, and the answer we get is 0.3 for the allele frequency for allele number 2. 30% of the genes, of the RFLP genes in that population, are allele number 2. A faster way to get the answer for allele number 2 is simply to subtract the allele frequency for allele number 1 from 1, since the allele frequencies have to add up to 100%. So 1 minus 0.7 would give us 0.3, a faster way to calculate allele number 2. In your book, we go through all of this material to illustrate what we mean by genotype frequencies and allele frequencies.